So, Mycobacterium tuberculosis is still the deadliest human pathogen in the world. On average, it'll claim the lives of 12 people during a 3-minute thesis presentation. The major obstacle for effective tuberculosis therapies is that our golden bullet against bacteria, antibiotics, are largely ineffective. The current treatment is a combination of 4 antibiotics for a minimum of 6 months. This is because Mycobacterium tuberculosis is an ancient superbug. It is the invulnerable Achilles of the bacterial world. And similar to Achilles, Mycobacteria have evolutionarily been dipped in rubber sticks to acquire an abundance of antibiotic resistance mechanisms. The goal of our lab is to understand how these mechanisms function in order to design ways to stop them. Specifically, my thesis is focused on a small protein called YB7. Mycobacterial strains lacking YB7 become susceptible not just to a couple of antibiotics, but several structural classes. These classes represent dozens of already clinically available antibiotics that can be repurposed to treat tuberculosis if we can find a way to stop YB7. Now, antibiotic-resistant systems have a high metabolic burden. They are a detriment to cell growth. And it is my hypothesis that to minimize this burden, proteins like YB7 are required to activate antibiotic resistance to combat antibiotic challenge. Using a variety of genetic, molecular biology, and biochemical tools, my work has uncovered how YB7 activates antibiotic resistance in Mycobacterium tuberculosis. Importantly, what I've shown is that we can combat this resistance by something as small as removing 1 carbon from YB7. This represents a unique, highly specific, and novel target to combat antibiotic resistance. Our goal now is to use my results as a model to design small molecule inhibitors of YB7 function. These inhibitors will open the door to new and desperately needed tuberculosis therapies by turning one of Mycobacterium's greatest strength into its Achilles heel.